And now I guess you have one more thing to show us, right?
Yeah. I will do the demonstration part of how you can visualize a protein in a 3D way. But
Okay,
before that,
so
you must know about some sites which are useful in this field, I guess. It is the RCASP site, which is a protein data bank. Like you can get the PDB file of any protein here. Um, Suppose I worked on human angiogenin, so you can get the PDB file of it here. When you are trying to visualize it in through PyMol, you need this PDB format. All the proteins have their PDB IDs as well, so you can download the PDB format through this site. That is the CSP PDB, and yeah, you can also see the faster format here directly. If you need the faster sequence, you can get there. You can copy the what are, you can understand what are the amino acids are there looking at the faster format, and then you download the PDB format, which I've already downloaded, so not going to download again. Now this is another site which is known as the PubChem.
So
Here
what
you
is
can
the difference between PDB format and this FASTA format?
PDB format is just like a text file which we are gonna use in this, uh, which this software gonna take, and it will give us the it will give us the structure. But the faster format tells you about what are the amino acids present in it, and if you need the nucleic, what are the um, peptides that are present in it, the triplicodons, and those you can.
Okay, that means in PDB format you are saying that means if there is tertiary structure, that type of information also we can find in the
Yeah,
PDB format.
yes the
Yes, and FASTA is giving only the sequence. Nah.
yes the
Okay, okay, thanks.
and we are gonna use this PDB in, like, there is a special software known as PyMol, through which we are gonna see the structures, which is gonna visualize it. And we know there are some sites like NCBI and Ensemble for the gene browsing, and Uniprot is similar kind of a site which you can use, similar like this RCSB PDB bank. And there comes the Swiss model. Suppose there a condition where you know the amino acid sequence but you don't know its structure. In that case, Swiss model helps you. Like I did the same thing. I copied this faster format, this amino acid sequence, and I put it in the Swiss model. So it has prepared me a structure. Like it can have this kind of a structure. So if there is a case where you know the amino acid sequence but you don't know its structure, you can use the Swiss model. Now coming to the primal part. This is a free software which we can use to visualize a protein. Basically, it's used to find out the protein ligand interaction and various other things. So, see, this was the PDB format I was talking about, 1B1I. As you see, you can see the 3D structure of it, which was encoded in this PDB format. Now, you see that there are some asterisks. So, these are some water molecules which are present around the protein molecule. So, we can just hide those, hide non-bonded. This is like some cheat codes which we used to use in the GTA Vice City or something like that. Here we can also use some cheats. You see that all the water molecules are now hidden. Now there can be many structures where we don't know where the ligand is, whether there is ligand in it or not. But since it's not that complex of a structure, we can easily say that it's the ligand. But there are very many complex structure. In that case, what you need to do is you need to go to the S part, which means the sequence, which will give you the amino acid sequence in the one letter code, and there will be break there, and that's the ligand which is embedded in the protein enzyme. Sorry, I said. Now what we are going to do is that we are going to select this ligand. We are going to rename it. Suppose we name it as our ligand. We are going to color it. Suppose We color it blue. So now we can classify which one is the ligand and which one is the other. Now the structure which is present in the front of your screens is basically this cartoon structures. These are not the only type of structures. There can be very many more structures. For suppose we hide this cartoon structure and we want to see the lines and the sticks. The same protein in the line form, but most favorable form is the cartoon form. Or you can also see the surface, like how does surface look like. If you want to see the protein surface, 3D surface, you can say it this way. But you again have to hide them again. Surface, lines, and we will go back to our cartoon. Now there are structures you can see in it. There are some helices, and the ones you see like the arrow-like structures. These are the beta pleated sheets. Now what we are going to do is we are going to label it like by the secondary structure. Now you can easily see like which one are the helices, which one are the beta pleated sheets. Okay, where did the ligand go? We have to color the ligand again, I guess. There is our ligand. Now we are going to use some more cheat codes again to see how the protein, how the ligand is attached to the protein, like how there is an interaction between the protein and the ligand. For that, we are going to use the cheat code. So sticks fires all within, and we have to write whatever the ligand name you have given, whatever the you have, like whatever the name you have given to the ligand. It can be anything you wish, so you have to write it here. See that the neighboring amino acids which are in the range of 5 angstrom. Now you might ask why I have written 5 angstrom. Basically, when in experimentally it was found that the hydrogen interactions that we have in a protein is basically from 2 to 4 angstrom, but so that's why we are giving it 5 angstrom, so we can find the hydrogen bonds which are present in it. And there are more bonds like sulfide bonds which occur in around 3 to 4 angstrom. So we are going to see whether these are the amino acids which are in the 5 angstrom vicinity. Now we have to label this manually. The ones which are close, you have to label them manually. But before that, we can do one thing, like we can hide the cartoon. Now you see that there's ligand and there are the neighboring amino acids which may be having interactions with the ligand. Now what we selected, and the thing which I was talking about in the slides about active sites, these are the active sites. Let's rename it as active sites. And let's color it too. Maybe let's color it green. Now you see the neighboring groups and the ligand. Now what we are going to do is we are going to find whether there is any polar interaction between them. Polar contacts to any atom.
So for the time being, you are just explaining, is there any hydrogen bonding interaction or not? Na?
Yeah.
Okay.
It's not capable to find all types of bonds, but it can find the polar interaction of the hydrogen bonds. Yes, you can find the hydrogen bonds. They are present in atom.
So which type of interaction it can we can find by this software?
Mostly the hydrogen bonds.
And the polar bond also.
Yeah.
Polar interaction.
Yeah.
Okay.
So you can see that with the ones which were in the vicinity, they have formed contacts. Like the yellow lines which you see are the hydrogen bonds, or you can say the polar bonds. Now you see that there are some bonds which haven't attached with anything. Like it's like they are attached to the air. But it's not the case basically. We have to show non-bonded. See those dots again. These are the water molecules. It means that they are hydrogen bonded with the water molecule. Now what we are gonna do is, let me select this. Where are the other hydrogen bonds? See, this is another water molecule, and this is another water molecule. So we are gonna rename it. Let's give it a name like active water, maybe active water. It's in the active type for active water, and we can show it in sphere form so that it's visible, and remove the other water molecules. Hide non-bonded. So that's basically whatever the polar interactions you had. There's a hydrogen bond between the water molecule and the ligand. There are three water molecules with which it's attached to it. Now to confirm more cases, like we can see like if there is any other group with which this hydrogen and this water molecule can form bonds. To do that, we will use the one cheat code again. I think it's preferable to remember, or you can write those cheat codes in separate way so that you remember it. So 6 is all, and 5 of active water. There's a new group introduced, which is in the vicinity of 5 angstrom, with which it's possible that it can form a hydrogen bond. Now, how do we check it? For that, you have to measure the distance. As I said, you recall that most of the hydrogen bonds that are created are between 2 to 4 angstrom, or maximum 2 to 4.4 to 4.5. So we need to see the measurement. So we create a new object. Now let it be the water molecule, and this part of the new chain which was introduced to us, 2.8. So yes, it's possible that it can form a hydrogen bond, and that's how you can check it. Like if you want to know that from that position of ligand to this tryptophan, if it can form a hydrogen bond, so we are going to take on it and take on it. 7.3. So basically it's impossible to form a hydrogen bond to that certain of a distance. So there will not be any hydrogen or any polar interaction between this tryptophan and the ligand. So that's how you check whether they can form the whether they can have polar interactions or hydrogen bonds. So this is a proper you can say that protein ligand interaction. We will remove the measurements. You can remove it from here. Now it's said that you need this kind of a pictures in the publications. For that, many of say that you have to do the display settings and all, but no need to do so much. You're gonna go to this draw, this ray slowly, and save image. Let's save it in music, I guess. Whatever the name we give. I will see in the water. This kind of images are mostly favored in the publications. See, these are the publication images you can have. Now I will also talk about the quaternary structure, which I couldn't show before. Well, in this PPT slide, it's difficult to show the quaternary structure. I will show it here. Like I have taken the PDB from the same site, RCSP PDB. We open desktop. So basically, this is the quaternary structure. This is a hemoglobin structure. The quaternary structure, you can have 4 chains in it. First, let us remove the water molecules which are present. Hide non-bonded. All the water molecules are gone. Now, how do I say that it has 4 polypeptides? It has 4 polypeptide chain. So you can color it by chain simply. Like use the chain, and you can see that there are 4 polypeptide chains, and they have interactions with each other, and they form the quaternary structure. 4 polypeptide chains, and if you see the sequence of it, it has 4 heme group. Like this is one chain. You see the sequence, and there are 4 heme groups. You see that there are 4 chains and 4 heme groups in it. You can manually color those heme groups too, like if you want. This is one heme group. This is one heme group. This is one more. We don't need to select this. There is one more heme group, I guess. Yeah, not this. So you can color this heme group too. So that's how you can see that there are 4 heme groups in it and 4 polypeptide chains. So that's how you can see the quaternary structure too. I guess that's how you see the protein ligand interaction through PyMol, and you can visualize any protein through it. I hope you could understand this tutorial.
One thing
Yeah,
that uh, like there are also like in case of like this heme group and with this sorry in this the porphyrin ring and that uh, other this protein chain, there are also like some direct bonding are there like with the 16 and other things. So how can you show that? Is
as
this can be visible by this pie chart? I sorry,
no,
by
the
pie
it
model.
can only show the polar bonds, and if there is a ligand interacted with it, it's not possible for PyMol to show every bonds that are present, like the bonds between 2 amino acids, the peptide bonds, or the covalent bonds. It can't show that.
Okay, 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 okay. So actually, so there is like Fe group now, and with this Fe group, there are
Yes,
like
yeah, yeah.
two peptide are like binding
Mhm.
with their stinent. Okay, fine. Yeah, very nice.
And if you want to know about the docking part, we can also dock molecules. Now, what is docking basically? As I said, I will use the ribonuclease. First, let me show you. Like these are the proteins which where we had something in it, right? We had some ligands attached to it. But if you say this ribonuclease enzyme, it doesn't have a ligand attached to it from the beginning. As you can see it from the sequence. To see whether there is a ligand present in it, the best way is to see the sequence of it. As in the sequence, you can see that there is no ligand attached to it. So we will try to attach a ligand to it. And that's what is indeed molecular docking. It is basically the interaction or the orientation which we will have between a protein molecule and a smaller molecule, which can be drug or which can be a ligand. So how they are going to interact and what will the orientation of it. So I will try to show it by doing the docking. I will
Can
dock.
you repeat what you said? Because there
Yes,
are red. Yeah, you said there are no ligand, but there are like red lines. And what is the
these
red?
water lines,
No, no, no,
the
not. I'm not asking about the sequence that you told last time that these gaps are actually represent ligand. So you are telling there is no ligand, then there are gaps. Are
I didn't say the gaps are not the ligands. These asterisk ones are the hydrogen bonds, and they can form hydrogen bonds with it, but uh, you can't find like in the, see, even in the previous ones you could find in the sequence there is him, you could find there is CIT in the first human antigen which I showed. If I have to show it again, there will be a CIT after the bond after the amino acid sequence. Here you can't find a ligand in it. These are just the water molecules around this ribonucleus, pancreatic
Oh, okay,
ribonucleus.
okay, okay.
Like, there is no ligand embedded in the structure. So I will try to attach a phosphate group in it. It said that the phosphate groups can attach to the ligands. They are better ligands to attach, but just for showing purpose, I will try to dock a phosphate molecule into it. First of all, you have to download the PDB files from the structure, from the sites, and you need a phosphate group from the pubchem. You can find any kind of drugs in it, or you can find any kind of ligand in it. So I downloaded the phosphate ligand. It's basically in the SDF form, but when you try to do docking, this SDF won't work. You have to convert this SDF into the PDB format. For docking, you need to have the PDB format, but it's nothing to worry. Yeah, anyone
Uh,
else?
Somo, can you kind of tell us more about what the PDB format is? Like, tell us as in, like, say I want to make a PDB format file from scratch by using Notepad, maybe. So, like, how would that look like?
I
Or
have
is it
no
even
idea.
possible?
I have no idea
Okay,
about it.
not a problem.
Like, it's a specific, like there is a PDF format. Like, it is a kind of a format. You can say that it has a specific
Okay.
format, PDB
Like for
format.
example, if I were to make a CSV file, then I know that CSV is comma separated value, so I would essentially be writing everything in a column-wise order where everything is separated by comma, like that. So in that context, I was asking.
Okay
You could
then.
maybe just try to open one of the PDB
Can
files
I
via
say
Notepad
something on
and
PDB?
Uh,
yeah, sure, Ritra.
The
well,
If
full
that's
form
fine,
of
so
PDB is that protein data bank. Uh,
protein data.
from where I am, we save their format, and especially the format is a structural format that give only the PDB. The protein data bank gives that format to to do the docking and forward things.
Okay.
I think Rishigupta, it's a, like an image like thing. Like with Notepad, it's difficult to produce an JPG image. Na? Like if you write in Notepad and
You
you
can't
We can also
do
consider
this
it that
on
convert it.
Notepad.
it has the codes present in between, and of with the help or the Autodoc softwares, we can use these codes to get a 3D visualization.
oh, okay, so I'm guessing it's kind of like a Fitz file in astronomy. So like a
Yeah.
Fitz file is kind of similar to that. So it is essentially a file where you can get spectroscopic data and then fit it into a curve. So I and also produce an image format. So I guess it is kind of similar to that maybe.
Yep.
Is it?
Maybe,
Okay.
Maybe.
maybe. I'm not sure about it.
Maybe. All right. Okay. Thank you. Understood.
So basically, I've downloaded all the files required for the docking. The first step of auto docking is preparation of the protein and the preparation of ligand. First, we will do the preparation of protein. I have my PDB format ready. We drop it here. Now we are preparing a protein for the docking. So what you're gonna do is edit. We're gonna remove the water. We edit. Then we are going to add. We are going to add the polar hydrogens. Then we edit again, and we give the charges in it. The most basic charges we add is the Coleman charges. So 8.0 Coleman charges had been added, and now we save this. in a PDB-QT format. Like after you have the PDB file, there's another type of file that is PDB-QT file. For the docking, you need that PBDQT file. So I saved this file before as protein PDB-QT. I'm not going to save it again, but this is how you prepare protein for docking.
Soumya, do you know what is PDB-QT?
No, brother, you wish to explain about it?
Okay.
If you want to explain, you can explain, brother.
Yes, PDB is protein data bank, and the PDB-QT is there's Q stands for the charges you added, and the T is the atoms you added on the PDB format. So it changes to PDB-QT, PDB to PDB-QT format.
Means just adding the charges and whatever the
And the,
changes we did, so
and
it
the
changes from
values.
PDB to PDB-QT.
Yeah.
Okay, thanks, brother. Now what we are going to do is, in a similar way, we can also prepare a ligand. Like, as I said, whatever we downloaded was in CDF format, so that won't work. We have to change this molecule into the PDB format, and we can use same software, that is the PyMol, for it. That is the phosphate molecule. You can save it like export molecule. You can name it. I did as K. Then you have to save it as the PDB file wherever you wish. So I had previously saved it as K2 again. So I'm not going to save. So that's how you convert a CD format, CDS format to the PDB format. Now again, when you do the docking, you drop it here, and that's the ligand you had. We are going to do the ligand. We are going to input, going to choose. It has been auto docked. It has been prepared, and now what we are going to do is save it as PDBQT. Now again, you have to save this file as a PDBQT. So I did save it as lig.pdbqt. Now these are the steps which we were doing at first: the preparation of the protein and the preparation of the ligand. Now what you're gonna do is bring we already had this file, so we wish to bring the amino acid 2 in here, like the protein we had and the ligand we had. So if you find the you can't find the ligand very easily because it's gonna be away from the protein. It's difficult to find the phosphate group in this big structure but it's going to be stay away from the protein structure because it hasn't been docked yet. So what
Yeah, do
we
it
have,
full screen. Yeah.
okay. So as I said, it's difficult to find the ligand present where is the phosphate group. But now we have what? Just wait a second. What we gonna do is like we are going to do the grid, and then we are going to choose our macromolecule. See the ligand which we while preparing the ligand we had already we did the preparations, but for the macromolecules we have to choose again. There are various subjects where we don't have to choose it again, but here we have to choose it again. 1fs3 We select the molecules, we say no. Okay, it should have been saved till now, but there's no option coming. Like after you do this, there comes an option. Now it means that your both ligand and the proteins have been prepared. Now what you're going to find is the grid box. Now you see here, that is a grid box. Now what happens is that here the ligand is gonna bind to the protein. Like there is a grid box, there are various coordinates given in which the protein and where this phosphate group will get attached. Till now, yeah, see, that's the phosphate group. As I said, that's going to be away from the protein. So it hasn't been docked yet, so it's going to be away from it. So that is the grid box which is formed where the where this phosphate group will and later it's going to attach there. Now what you need to do is that you have to make a grid file. Sorry, you don't need to form a grid file. You can have it from here. You can use the output grid from and you can have the grid. It will be saved as txt format. Now what you need to do is that using this um, these coordinates, you have to again form a config a log file. Sorry, a configuration file, which you have to do in the exact same way which we have written. Like there should be receptor ligand, then there should be center x, center y, and the coordinates which we are given. I prefer not to change those default things which we are given. Like this energy range must be 4, this exhaustiveness should be 8. And why this is 8? Like this is gonna do the docking 8 times in an exhaustive way. That's the default things which we are given. And the size x, size y, and size z, if you don't know it properly, don't change the default values. But you were giving the grid box coordinates, you can also change, by the way, but I prefer not to change. And you must remember that the center spelling is center. The mistake I did in the beginning was I was writing centre, so that was not working well. So that wasn't working at all. So you have to write the spellings and the everything in a correct way. So you should know this exact configuration. Now what are the things we have done till now? We have prepared a protein, we have prepared a ligand, then we have made a grid box to know where the ligand is going to interact with the protein. Now we have prepared a grid file, and uh, we have a grid file, we have a configuration file. Now we will go and do the docking, the main part. Basically you can't find a specific app to do or a software to do that. You have to use the command prompt for it. So we change the ed refers for the change directory, and then we go to the intern and we give the input file, like where we have saved all our pdb requirements, like the grid file, the configuration file. That path you have to copy. I came to a different again. You have to copy that path, and then give a space and paste it. Now this is a format which you should remember, because most of the problems we face later on occurs at this position. And I prefer you to save the location of this. Like if you download it from this sites from a specific site, you will find that your Autodoc Vina or the exe file where to save the exe files is, you just save this somewhere else, because this is the most notorious thing to find again and again. You save it. It is the path where you have saved your Autodoc Vina exe files, like after downloading it from the net. To go back to the command prompt, will be vina.exe, and what we are gonna do, some space dash. Like we are using all of the things which we have made till now, and we are gonna do the auto docking part. Receptor. I had saved the protein as protein.pdbqt, pdbqt. You have to remember the names with which you have saved the protein molecules. Like if you name it as something else, you have to write the exact things. You can't change it here. Then comes the ligand file which we prepared. Ligand is, I saved it as lig.pdbqt. Then we made a configuration file. So we are gonna do config.txt because we saved it as txt file. Then there's gonna be the log file, log.txt. See, basically the log files, whatever thing which is running in this Autodoc Vina is the log file. You don't need to worry about it. And then output.pdbqt. So see now, let's see if it runs or not. Output will be also in the pdbqt format. We need it in the pdbqt format. So yes, the Autodoc is successful. Like we will get the final output file. It will be saved in there, but these are the scores. These are the affinity scores you are gonna have. The lower the affinity score, it's more better for the ligand, and it will be docked better. So as I said, that just simple phosphate isn't a better ligand, but better ligands will have it around 6 or 6.7 or 7 or more than 7. So lower the affinity score, more it's better, it's more better docked. Now you can see that the output file in the pdbqt format, it's prepared for you. Now if you want to see that whether this docking has been completed or not, we are going to use PyMol again to make it sure. Like you need to see that whether it has been docked or not, to see whether the ligand has been attached to it or not. We're going to do this output pdbqt under the PyMol, and we are going to bring this into this. As you see this, it's somewhere close to the protein, but it's not completely attached to it. The better the ligand, the more better it is attached. So that's how you do the docking. If you take more phosphate ligands, then there's a chance that it will be better attached to the protein. And as I said before, RNA had nothing in it. Our ribonuclease enzyme didn't had any embedded ligand to it, but now this phosphate group is somewhat near this protein, and you can see that it's close to being docked, but it's not completely docked. That's why I said lower the affinity score is better to have. See, and that's the hardest part, to find to find the appropriate ligand. That's the toughest work, so that it can get embedded into it. So whatever the total things we did in docking, we did the preparations of
So
protein
we
and
have
the
to do
ligand.
like trial and error with this
Yeah.
ligand.
Exactly. You have to study about the protein. What are the expected ligands you can have so that it attaches? That I studied about RNA ribonuclease, so it said that it can attach phosphate based ligands. So just for showing this docking for this, I did that I took the phosphoryl group and tried to dock with it, but it's not the best of the ligands to attach with it. So better the ligand, more it is docked, more is the low affinity score, and it's better. Like it will be directly embedded into it. You have
Okay.
to do hit and trials. You have to study about the protein, and you have to see what are the ligands we can attach to it.
Okay. Okay.
So I guess that was all for this interactive session.
Yeah. So very nice talk, Shomo. Thanks for that.
Yes, thank you so much for the nice, wonderful talk.
Thank you, guys. My pleasure.